Hi, my name is Laura Gunsalus, and I'm a third-year graduate student at the University of California, San Francisco. I first became really excited by problems in regulatory genomics as an undergraduate at Carnegie Mellon, where I found it a really natural way to combine a love for genomics with fun algorithmic solutions from computer science. So since then, in my PhD work, I've been really fascinated by how the three-dimensional conformation and folding of the genome influences what genes are transcribed and when. So I'll walk through some of my ongoing work in applying interpretable deep learning methods to interrogate how the structure of the genome influences its function. We often think about DNA as a long string of nucleotides, but in fact, it's tightly compacted into the nucleus of every cell. If you were to stretch out DNA from just one of your cells, it would span over 2 meters in length. New imaging techniques allow us to visualize the genomes of individual cells for the first time. This allows us to ask questions like, what's the difference between cells where genes are being actively transcribed and cells where genes are being turned off? Is there anything different about their structural genome conformations? We're interested in the predicting relationship between 3D genome conformation and gene expression using a graph neural network. But to do this, we'll need a way to represent the stated modality. We'll use a graph where every node represents a genomic bin and every edge represents the distance in nanometers between two bins in 3D space. We could also include information about what's happening in the cell, like distance to nuclear speckles or nucleoli or the nuclear lamina. If we are to divide our data set into cells where the genome is highly compact versus cells where the genome is open, our graph neural network is able to easily distinguish between the two cases. We received around 95% accuracy on the validation data set. But if we actually try to predict transcription, we do much worse, and we get only around 60% accuracy on the validation data set. This might be because of the incredible amount of stochasticity in single-cell data sets. We often think about these data sets as individual examples, but instead we're actually capturing snapshots from what in real life is a dynamic pose of a very fluid Cromatin structure. Because of this, we might not be able to accurately predict transcription from the behavior of individual cell snapshots. With that, I'd like to thank the incredible amount of support I've received from my two labs, as well as the ARCS Foundation.